Hello students, welcome to part 7 of chapter 11 Enhancement in Food Production. Today we are going to discuss how microbes in human welfare are used for production of hormones and enzymes. First we'll discuss about gibberellin production. What is gibberellin? Gibberellins are groups of hormones. They are plant hormones, phytohormones. They are mainly produced by higher plants, even by some fungi, and they promote growth of the plant by stem elongation. The first gibberellin was isolated and crystallized by two Japanese scientists, Yabukta and Sumiki, in 1938 from rice seedlings which were infected with fungus Gibberella fujikuri. But it was discovered before them by Kurosawa. Kurosawa was working in a field. He saw that some of the sugarcane stems, as you can see in this picture, they started growing too long. There was too much, the height increased too much. He studied carefully and he observed that these sugarcane plants, they were infected with a fungus gibberella, and some chemical was secreted by that fungus, and he called that chemical as gibberellin, which resulted into the stem elongation. This disease is also called as the Buchan's disease or foolish seedling, because the seedling becomes foolish and start growing too much in height. Now, about 15 types of gibberellins have been isolated from Gibberella fujikuri, whereas more than 126 types of gibberellins have been isolated from fungi, bacteria, and higher plants. Let us see what are the uses of these gibberellins. So, it is used in stem elongation of the crop plants. It induces parthenocarpy in pseudocarps like apple and pear. Parthenocarpy, that is production of fruit without fertilization, whereas apple and pear, these are pseudocarps, but parthenocarpy can be induced in them also. They help in inducing flowering, breaking the seed dormancy, increasing the size of the fruit, and enzyme induction. So these are uses of gibberellins. Let us see about how enzymes are produced and what are enzymes. Enzymes are biocatalysts. They initiate or accelerate the rate of chemical reactions or biochemical processes occurring in living organisms without participating in the chemical reactions. Many different types of hydrolytic enzymes like the starch degrading, fat degrading, protein, pectin into simpler compounds are known. Enzymes like amylase, papain, protease, pectinase, streptokinase, lipase, all these are industrially or commercially produced. What are the uses of enzyme in industrial processing? Where are they used? So basically, if you see, they improve the fabric quality in textile industry. When a fabric is formed, initially it is pale yellowish in color, not so smooth and fine. So the quality is enhanced by using enzymes. Biomechanical pulping and bleaching the paper industry. The paper, when produced initially from the bamboo pulp or wood pulp, it is pale in color. It is bleached and given very white and fresh color using enzymes. Lipases are also used in detergent because to remove the oil stains, these lipases, they will break down the fat or the oil particles which have been settled on the cloth and remove the oil stains. They are used to produce desirable flavor of cheese and butter. They're also used to obtain desirable sweetness of confectionaries, that is chocolates, animal feed, soya bean milk, food gums, etc. Enzymes are used in the production of beer and wine, and they're also used in the cosmetic industry. So these are the different uses of enzymes in various industries. Question can be asked on 2 marks on this. Let us see some very useful enzymes. First we'll see the enzyme, then their sources and how they are useful to us. So enzymes like streptokinase, also called as tissue plasminogen activator. Second enzyme we'll discuss is about statins, and the third enzymes we'll discuss are cyclosporins A. I repeat, streptokinase, statins, cyclosporinase. Streptokinase, also called as tissue plasminogen activator, thus they are obtained from bacterium Streptococcus species. They are having a fibrinolytic effect. Fibrinolytic, it does not allow the fibrinogen protein in the blood to convert into fibrin. If no fibrin is formed, clotting will not take place. And where it is used as a clot buster for clearing any blood clots in the blood vessels. So basically, it will clear all the fibrins produced. It will break down the fibrins and thus clear the clots in the blood vessels of the patients, which may be very dangerous and cause heart attacks. Thus, streptokinase is very, very useful to heart patients having blood clots. Statins are obtained from yeast Monascus purpureus. These statins, they are blood cholesterol lowering agent. So interesting. These statins, they compete with the enzymes who are producing cholesterol. So suppose this is an enzyme who is going to produce cholesterol. These statins, they will compete and they will not allow the enzymes to produce cholesterol. So obviously what will happen? The level of cholesterol will go down. Synthesis of cholesterol will be reduced. Thus, it is very useful again for patients, especially who are suffering from coronary artery diseases or heart diseases. Third, cyclosporine A, it is obtained from a fungus Trichoderma polysporum, and it is a boon. The cyclosporine A is a boon for in case of organ transplantation, because it is used as an immunosuppressant in case of organ transplantation. Now suppose this is a donor, this is a recipient, this donor is giving its kidney to the recipient. The body of the recipient somehow tries to reject the kidney because it is a foreign substance by its immune system, but this immune system is suppressed by cyclosporine A, so that the recipient will easily accept the kidney. So in not only in kidney but any organ transplantation, cyclosporine A can be used. Let us see different names of enzymes and their microbial sources. Invertase, it is obtained from Saccharomyces cerevisiae, that is yeast. Pectinase is obtained from Sclerotinia libertine or Aspergillus niger. Lipase is obtained from Candida lipolytica, and cellulase is obtained from Trichoderma conigi. You have to learn these names by heart. So invertase, pectinase, lipase, cellulase. Invertase, Saccharomyces cerevisiae, yeast. Pectinase, Sclerotina libertine, Aspergillus niger. Lipase is obtained from, easy to remember, lipolytica. So Candida lipolytica, and cellulase is obtained from Trichoderma conigi. So students, in next video now we will discuss about how microbes are used in sewage treatment, and we are finishing here. Thank you.